The U.S. Department of Energy is cleaning up the Oak Ridge Reservation of residual hazardous and radioactive contamination left over as a result of decades of nuclear energy research. Much of that work has been influenced by the Oak Ridge Site-Specific Advisory Board, a citizens panel that provides independent advice and recommendations on DOE's cleanup operations. You're welcome to attend the meetings and be part of this important work.
While they're getting seated, I want to welcome everybody to our April meeting. It's good to see the room full for a change, and online is full too. Look at that. So there's a full screen. It's kind of nice to see everybody back. I'm gonna go into comments from Melissa. I'm gonna let you start first.
All right. Well, good evening, everybody. Thank you for being here. We have a couple of updates for you since our March meeting. Our biggest update is that our contractor, UCOR, has recently signed an agreement with the National Labor Organization, North American Building Trades and Unions. That will boost pay and benefits for workers in Oak Ridge. The agreement offers a 20% wage increase over a 3-year period, plus paid holidays and a retention bonus. We had been having a little bit of attrition due to some heavy competition with other projects, and this will give our program, help us attract and retain some of our best workers. We're the first EM site to do this, and it helps set a template for other sites to follow. Our crews have finished a major infrastructure upgrade project for our waste treatment system at ORNL. The project replaced 2 miles of piping on the liquid and gaseous waste operations, which is essential for the Oak Ridge National Laboratory ongoing operations. It involved a lot of work and coordination, including more than 5,000 hours of welding. Workers completed the project $900,000 under budget and 3 months ahead of schedule. So we're very proud of that. Lastly, we wrapped up early site preparation activities for our EM disposal facility. We're now moving full steam ahead on the second phase of the project, which is the groundwater field demonstration study. Thank you for your attendance again. And with that, I'll turn it over to the next speaker.
Roger. Mr. Petrie, that would be you.
I don't have anything to add, but always happy to answer any questions.
Thank you. Is Samantha? Hey, look, Samantha. You got me tonight. Samantha, do you have anything that you want to tell us about?
I'm excited to announce that PFAS was added, an MCL level added to the Safe Drinking Water Act with an MCL level today. So that's exciting news.
Well, you really have surprised me by being here in person and not on the screen, but welcome. Thank you for coming this evening. We really appreciate it. I know, right?
Yeah.
Are you almost settled? Getting there.
Christoph?
I have no comments, announcements, other than being happy to be here and ready to answer any questions.
And we always appreciate you being here. We will now have our our introduction of our presenter.
Teresa Matthews is the senior scientist and group leader for the Biodiversity and Ecosystem Health Group at Oak Ridge National Laboratory. The research is focused on understanding how ecological interactions can affect the biochemical cycles of nutrients and contaminants in aquatic ecosystems. She received her PhD in coastal oceanography from the School of Marine and Atmospheric Sciences at Stony Brook University in New York in 2007 and joined Cornell in 2009.
Thank you.
So thank you all for inviting me here to listen to all of the exciting research that we have going on in Oak Ridge National Lab focused on mercury. So I'm going to be talking to you about just broadly about the aquatic ecology research that we have, specifically as it focuses on mercury remediation technology development. Our biological monitoring program turns 40 next year, and the mercury remediation technology development program is in its 10th year this year. So I won't be able to get into all of the details. I'm just going to give you like a broad overview, but if you want to have discussions, I know that we'll have time to discuss afterwards. And I hope that all of you will visit the aquatics lab.
No.
So before I get started, I just want to say that mercury is a very complex subject. Mercury remediation and contamination is very complex. We lean on, like I said, 40 years of history and expertise at the lab, but we also have a lot of partners. So we work across the Oak Ridge complex. We have support from UCOR, RSI, Y12, across the DOE complex, other national labs. We are not here to pick any specific, our favorite technology to clean this problem up. We are reaching out to experts across the world in mercury research. So we work with other contaminated sites. So the South River Science team, we work with other industries. Basically, we want to get the most cutting edge science to apply to our problem here in Oak Ridge. You'll see that we work with a number of universities. We actually have some students that come from some of these leading labs across the country and then go on to be our collaborators as they move on in their careers. So we're really building a network really of mercury experts across the world to pull from. So I will talk in general about mercury as a global pollutant. So any remediation technology development that we develop here in Oak Ridge is applicable across the world because mercury is such a global pollutant. I'll talk about the mercury problem in Oak Ridge. There are some very specific things to Oak Ridge which make it particularly interesting and particularly challenging. And then I'll talk about mostly the strategy for our remediation technology development. And then again, as if you want to get into more details, we can do that in discussions and then talk about future directions. So mercury is one of the most interesting elements to study because it can undergo so many different transformations in the environment. It can be oxidized or reduced through photooxidation from the sun. It can bind with dissolved organic matter in aquatic ecosystems. It can volatilize and travel for many hundreds, thousands of miles before it settles back down on the earth. So there's a lot of interesting things to study with mercury, but the most important transformation that I'm going to focus on today is the methylation of mercury, which happens in aquatic ecosystems. Mercury can be methylated to an organic form called methylmercury, which looks like an essential amino acid. And so it's readily taken up by cells and then not given up. The cells don't give it up. So it tends to bioaccumulate in aquatic organisms. And so our biggest dose comes from eating contaminated fish. Because of this, because mercury is transformed in the environment, our exposure is affected by aquatic ecosystems and risk and toxicity. Also, how we remediate it is going to be very different based on these different forms. So keep those things in mind as we talk about Oak Ridge. Oops, going backwards. So mercury is one of the only metals that is known to biomagnify in aquatic ecosystems, and that means that it becomes more and more concentrated as it moves up the food chain to fish, and that's specifically true of methylmercury. So as I mentioned, our biggest dose of mercury comes from eating contaminated fish, and for this reason, EPA gives us a guideline not only for mercury in water, but also a fish tissue guideline. So we need to comply with the fish tissue guideline, and that's 0.3 micrograms per gram in fish fillet. That fish tissue guideline is considered to be a more accurate indicator of exposure and risk. But what this means is that because mercury biomagnifies, even low concentrations in water can lead to hazardous concentrations in fish. Oops, I'll get this by the end of the talk. So mercury contamination is widespread. This is kind of a dated slide, but I just like the way that it shows a number of different things here. So on the left-hand side, you see the river miles that are contaminated by mercury with respect to all other contaminants, and the lake acres on the right-hand side. And so you can see that mercury contaminates more watersheds than any other contaminant across the United States, and that number has increased over time. And the bottom map, anything that's shaded in green is a state that has some kind of advisory for mercury, and you can see that mercury is a very widespread problem. And it's not only in the United States, so this is global mercury emissions. You can see that the United States is not the biggest emitter of mercury. When you look at the emissions in the United States, you can see that coal combustion is one of the biggest sources of mercury to the atmosphere, but there are a lot of other sources of mercury. In particular, in other areas of the world, mining can be a very important source of mercury. But we're here to talk about Oak Ridge. So the world's stockpile of mercury was brought to Oak Ridge in the 1950s and 60s. Before the Manhattan Project, Oak Ridge was an agricultural area. And while we don't have concentrations of mercury in the environment from that time, there are models that have been used to predict concentrations, just background concentrations at that time. And so concentrations in this area in the Southeast were very low. So we expect that there weren't high concentrations before mercury was used in the 1950s and 60s. And here are some cool pictures. I wish that we could see these better, but these are really cool. So on the top, these are the mercury flasks that are arriving at Y12, where mercury was used in the 1950s. You can see some pictures of the mercury facility unloading flasks. And then that bottom picture is from K25. Was the largest building in the world. And you can see that there's a number of different columns. That's where the mercury was pumped to separate the isotopes of lithium. And about 11 million kilograms of mercury were used during that time. And the processes were relatively clean in terms of industrial processes. About 3% of that mercury was lost to the environment. But as we learned, a little bit of mercury goes a long way. And so we're still dealing with the consequences of that. So during the 1950s and 60s, spills and releases of mercury contaminated the creeks, floodplains, and downstream sediments. This is just showing that most of those spills happened in the 1950s and 60s. And since then, mercury remediation has focused on source control, so controlling sources of mercury to the creek into the environment. So water treatment systems, sewer relining, pipe rerouting, soil removal, those kinds of things. I'm part of this talk. So this is East Fork Poplar Creek. This is the creek that runs out of Y12 into the city of Oak Ridge. It is the largest stream, the longest stream on the Oak Ridge Reservation, so about 25 kilometers long. It runs out of the west end of the Y12 facility and then into the city of Oak Ridge. Unfortunately, the font is pretty small on the map, but basically because it's 25 kilometers long, the sites that we study are named with the designation EFK, which means East Fork Kilometer, and it starts at 25 and goes down as you go downstream. As I mentioned, mercury use in the 50s and 60s resulted in contaminated soils, and there's been a lot of actions taken. This is another view. I'd like to show this map. This is a map that was created fairly recently, and it shows how the creek is affected by the land use. So let's see, so this is the creek here, East Fork Poplar Creek. It runs through the Y12 facility into the city of Oak Ridge. So you can see that anything that happens within the Y12 facility, the flow is really regulated by all of this, the complicated storm drain network. Most of the water that goes into the creek is chlorinated processed water from the Y12 facility. All of that affects how mercury is going to be speciated and transported off of the DOE facility. Then the creek runs into the city of Oak Ridge. And so you can see that there's a lot of development in Oak Ridge, and a lot of the development can also affect how sedimentation happens, how erosion happens in the creek, and then therefore how mercury gets transported. I'll highlight to you here that there are two areas that we identified with having high concentrations of mercury in the stream banks. The other areas have been remediated, but there are two areas that we're focusing on for our efforts for remediation in particular. I also just wanted to mention here that our research for remediation really pulls on all the expertise of Oak Ridge National Lab. So we are collaborating with the Office of Science, and they're really focused on very fundamental science related to mercury. So understanding the genes that were responsible for mercury methylation, those discoveries, like discovery science happened here at Oak Ridge National Lab. We've made some really big breakthroughs. We're using some of that science to apply to this problem to clean it up. Most of the work that I'm going to be talking about is from the on the OREM side, so mercury remediation technology development, but that also pulls from our 40 years biological monitoring program, so compliance studies. So it's a rich database of knowledge. So as I said, the biological monitoring program, we have 40 years of data on water quality and bioaccumulation. So we measure mercury concentrations in the water and in fish. We're targeting fish that are potential for human consumption, so larger fish. We also collect water samples for toxicity testing to make sure that we take the water samples back into the lab and expose them to lab organisms under controlled conditions to make sure that everything can live in there. And you'll get a chance to see that when you come to visit the lab. And then we do field studies at various sites to make sure that the communities that are living in the creek are diverse, because a diverse community means that we have very good water quality. We've tracked this over the past 40 years. This is just a summary of all the actions taken. Most of the actions have been taken at Y12, and most of them have been taken to address the mercury problem over the years. And they are in different categories. So water treatment systems, things like storm drain, cleaning or rerouting, flow changes or reroutes can affect mercury concentrations as well. Soil removal or stabilization or chemical additions. All of these things have been done over the past couple of decades. You'll see that in 2019, there was the construction of the mercury treatment facility began. A number of these things have been really significantly affected mercury concentrations in the stream. So here I'm showing aqueous mercury concentration. So mercury concentrations in water at station 17, which is our monitoring station in the creek, just as the creek leaves the Y12 facility. And you can see that in response to all of those actions that I just showed you on the previous graph, the mercury concentrations in water have decreased significantly over time. However, the fish tissue concentrations at that same site have not responded accordingly. So in the beginning, in the late 1980s, and oops, did something. I got out of presenter mode. Oops. Okay, I'll still talk while she fixes my mess. So in the beginning of the 1990s, the fish tissue concentrations were decreasing in response to aqueous concentrations. But after a certain period in the 2000s, we don't see a one-to-one response. The fish tissue concentration stopped responding.
Sorry, I am like, no.
It's my fault.
Yes, it's hard to, but it's very difficult screen to read, guys. There you go.
And you can see that the light blue dots are the fish tissue concentrations, and those are above the dashed gray line. The dashed gray line is the EPA tissue criterion. So we are still above that criterion. So that's at the upstream location, station 17, but let's look at how things change as we move throughout the creek. So as we're moving from left to right, we're going from upstream to downstream in the creek. And the blue line is water concentrations. So you can see that water concentrations are higher, closer to the Y12 facility, and decrease as you move downstream. The red line is our fish tissue concentrations. And interestingly, the fish tissue concentrations actually increase as you go downstream. And the reason for that is because, as I mentioned earlier, it's methyl mercury that is actually controlling how mercury gets into fish. So if you compare total mercury and methyl mercury concentrations, I'm going to try and not mess things up again. The dark blue line here is total mercury concentrations, and those decrease as you go downstream. So a source by the Y12 facility that decreases with dilution and as you go downstream. But as you go downstream, the creek starts to get more natural. It's not a storm drain network that's feeding it. There's sediment and biota that live there that can methylate the mercury. And so aqueous methyl mercury concentrations increase as you go downstream. And you definitely see that signal in the fish. So on the right-hand side, we're looking at two different species of fish. On the top, we're looking at forage fish that eat algae. Those fish, the concentrations are higher upstream and decrease downstream, again, because they're more related to what's going on in the water because they feed on algae. But the fish that we care about in terms of like our guidelines actually are the concentrations increase as you go downstream. And this light blue bar here within the total bar, this is the proportion of total mercury that is methyl mercury. So you can see that that increase as you go downstream is really due to methyl mercury concentrations. So what do we do about this? As we are going downstream, like we're decreasing mercury concentrations over time and like as we go downstream, but our mercury concentrations in fish either are not responding or are increasing. So how are we going to handle this? This is a paper that Dick will remember very well. What we did was we plotted on the top-hand graph, the top graph shows aqueous mercury concentrations and fish concentrations where we had water concentrations and fish concentrations at the same time. So water concentrations are on the x-axis and fish tissue concentrations are here. And what you can see is that the relationship between water and fish is not linear. And that's why when we were decreasing mercury concentrations from these elevated concentrations to here, we weren't really seeing a response in fish. It's not until you get to the lower aqueous concentrations that you start to see this decline. So this tells us that maybe some of the targets that we were aiming at previously might not be successful at decreasing the mercury concentrations in fish. So are there different ways that we can look at this? We know that it's methyl mercury that's controlling the fish. As I said, at Oak Ridge, we had this discovery of the genes that were responsible for mercury methylation, but we don't have the buttons to turn the bacteria off that methylate mercury. So are there other things that we can do? Are there other best management practices that we can use to try to decrease the methyl mercury concentrations? So that's when about in 2014 is when the remediation technology development program began. And the reason why is because there was regulatory pressure to address the mercury stream exceedances, both the water concentrations, the flux of mercury out of the DOE facility, and then also the fish tissue concentrations. And over time, because we've been studying the site for so long and we're understanding more and more about the sources of mercury, there was an increased understanding of the importance not only of the Y12 facility. So some of the actions that were taken were in the Y12 facility, but we also have diffuse legacy sources as you go downstream. And so if we're not addressing those, we're not really addressing a lot of the problem. Again, it's a complex system. We're probably going to need a watershed scale systems approach. It's probably not going to be one solution at all of the sites. And so we started off with an extensive literature review, again, reaching out to mercury scientists across the world, other mercury contaminated sites. We know that there's a desire to avoid a large scale removal of downstream soils and sediments. That's really disruptive. Again, people live in that area, and then also it's just disruptive to the environment. It's not really doable at such a large scale. So are there ways that we can enhance natural resources by leveraging ORNL's basic science capabilities? This is basically saying the same thing, except we started off this program really by doing a detailed field characterization. If we're trying to start doing these manipulations, first we have to understand really where we are now. So we did an extensive field characterization for the first couple of years of this project. As we're moving on, we're trying to pull all of the data together to try to use some predictive tools. And if any of you are engineers here, you know that probably the most complex problem that you can deal with is scaling up. So we can prove that something is effective in a test tube, but then scaling it up to a real world system. These are things that are very challenging. And so we're using the capabilities of Oak Ridge National Lab to make that transition. So the strategy focuses on developing technologies to decrease mercury in fish. And this came out of a USGS. They have a very strong focus on mercury science. So there are 3 key factors that are recognized to control mercury concentrations in fish. First, it's the amount of mercury that there is. That's fairly obvious. Then it's the conversion of inorganic mercury to methylmercury and the bioaccumulation of methylmercury through the food web. And so this program was designed to address those 3 key factors. The first 3 tasks that you see here are specifically aimed at those 3 regulatory drivers. First is decreasing mercury source inputs and flux. So we focus on soil and groundwater source control. The second task focuses on decreasing mercury concentration and methylation. So we're focused there on water chemistry and sediment. And then the third task focuses on the ecology, so decreasing mercury bioaccumulation. Again, because scale is such an important factor here, you all will get to see some of the upgrades that we did to the Aquatic Ecology Lab so that we could use it as a test bed for some of these technologies. And then, as I said, we're collecting a lot of data, and so we're trying to integrate all of that data to be able to make predictions in the future. So reducing mercury flux. I mentioned that we have identified some areas, some stream banks in the stream that have very high concentrations of mercury. You can see that in a stream system, the flows can change very widely from base flow conditions to storm flow conditions, that the stream banks can fill up, and that leads to erosion of these high mercury contaminated areas, leading to the most elevated fluxes happen during these storm events. So we're trying to focus on those events. So at the beginning of this program, we did a detailed field characterization. There were hundreds of samples that were taken throughout the creek. The sediments, the stream sediments, the floodplain soils, the stream banks to characterize where we were. This had been done in the 1980s, but hasn't been done since then. And we know that the concentrations have decreased significantly since the 1980s, but there are still some areas. You can see these areas where there are high concentrations of mercury that we can focus on that could be quick wins if we can address those problems then and stabilize those mercury inputs, then we can significantly reduce mercury flux to the stream. So we're focusing on those areas and we're measuring erosion at those areas. So we use LIDAR, which gives us very accurate measurements of the stream volumes. And we are to the point now where we can measure how much erosion we have and therefore how much mercury is sloughing off of those banks over very short time periods. We're not at the point where we could predict from one individual storm, but we're getting very accurate measurements of the volume that is coming off of these banks. And so we can estimate how much is coming off of the banks, and then what is the solution to that? We're actually proposing ways to stabilize those banks, the ones that are susceptible to mercury erosion. We're also doing lab studies where we are using amendments, so sorbents, to add to those mercury contaminated soils to see if they are effective at removing mercury from the soils. So here, this is an example of the setup, which you'll get to see in the lab, where we have East Fork Poplar Creek soils. We have water that flows. So we measure the mercury in the water before it goes through the soil. The water goes through the soil. We measure it after it comes off of the soil. So we know how much mercury came off of the soil. And then we put it through a sorbent. And so we know the mercury before it went through the sorbent and after. And so we can calculate the efficiency of removal of each of these sorbents. So this is mercury that came off of the soil. This is the mercury that came off of the sorbent, and that's the percent efficiency of that sorbent. And so we've done a number of these studies over the years to look at which sorbents are most effective at removing mercury from the soils. There's also a factor of cost. So some of these are like designer sorbents that we're using like material scientists at Oak Ridge National Lab to get the most. Again, doing something in a test tube is really easy, but then how do you scale that to the creek? So we've done some field deployments. This is where we've taken some of the most promising meaning it sorbs a lot of mercury and it's not a designer sorbent, like we can actually afford to deploy these things. We put them out in the creek. These are some students that we had to deploy these, we call them sorbent coupons. we deployed them in the stream banks and in the stream sediments and kept them there for a period of a year. This is a very rare kind of study that we can track things over a year. So here is where we're looking at the soils. This is an example of where we did the same thing in the sediments. After a period of 3 months, 6 months, 9 months, and 12 months, we collected those samples and analyzed them. And the interesting thing is that some of these sorbents actually continue to sorb mercury after a period of a year. So that tells us that we can keep these things deployed. I don't know if we went longer, if it would continue to sorb mercury, but these things are effective for long periods of time. Now, we're having to test these things in the lab as well, because we don't want to do something in the field that's going to adversely impact methyl mercury. So some of these things have organic fractions on them. And so we don't want to sorb all the mercury. Then we have a hotspot of mercury with organic matter for bacteria to methylate. So we're doing long-term experiments now in the lab, not only to look at inorganic mercury sorption, but then to look at the generation of methyl mercury. And you'll see some of that when you come visit the lab. So this task 2 is focused on water chemistry. And are there things that we can do to affect the water chemistry? I will walk you through this. We knew that we did not have a very good handle on on discharge in East Fork Poplar Creek, because we were estimating discharge to be higher at sometimes in the upstream sections than in the downstream sections, which doesn't add up. And so if we wanted to, so that would lead to bad estimates of our mercury flux, because basically we need to get the discharge of water that's coming out and the concentration of mercury. So at the beginning, again, similar to the sediment that I was talking about at the beginning of this project, we put gauging stations out at our 2 different sites so that we can get very accurate rating curves to be able to calculate accurate discharges. And here we're looking at, on the top, we're looking at total mercury. On the bottom, we're looking at methyl mercury as you move downstream. So this is a little bit complicated, but I will walk you through it. This is at East Fork kilometer 23.4, so station 17 at our upstream site, and this is moving downstream throughout the creek. The yellow is particulate, so any mercury that is associated with particles, suspended solids and such, and the blue is the dissolved mercury. And what you can see is, so these numbers here is the discharge, so the flux of mercury coming through these sections. What you can see is 20% of the mercury flux is happening in upper East Fork Poplar Creek, but 80% of the mercury flux, total mercury flux, is coming from downstream sources. And most of that is particulates. Methyl mercury is a different story. Very little methyl mercury, 3%, 4% is coming from Y12. And the majority of methyl mercury is coming from downstream. So now we have very accurate measurements of flux of total mercury and methyl mercury, both particulate and dissolved. Again, how we decide to remediate at different sites is going to be dependent on how mercury is speciating. So it was very important to us that we get accurate measurements of this flux. For the interest of time, I'll just skip over this, but it was another example of flow and mercury. This is showing an example of how we are altering water chemistry to reduce the flux of mercury. This is in the Y12 facility itself. In the Y12 facility, mercury is mostly inorganic mercury. There's actual beads of mercury that are in pipes and storm drains. You can see this is a bead of mercury here in one of the pipes. And as I mentioned, most of the water comes from Y12. It's chlorinated water, and we then use chlorinating agents to take the chlorine out. The chlorine and those dechlorinating compounds actually act to dissolve some of these mercury beads and then make it more available for transport downstream and for methylation and bioaccumulation. And so we actually proposed to use different dechlorinating agents. So this is an example where we did a study in the lab to look at, we took a mercury bead in a vial, and then we looked at how quickly that the mercury dissolved in the water. So this is the black line here is showing the mercury that came off of that mercury bead into the water over time. The red is sodium sulfite, which is a common dechlorinating agent. And the blue is what we were proposing to use. So basically it's vitamin C. And so by using a different dechlorinating agent, we got less mercury dissolving from these mercury beads. And while sodium sulfite can cause anoxia, which can harm fish, basically nobody has ever said that too much vitamin C has hurt anybody. So we're using friendly compounds that are actually helping the mercury speciation in the Y12 facility. Now I'm just showing you an example. I mentioned I can't get into all the details. This is just an example of something that has been used in Y12, is being used right now. But we are also using some of these same techniques to alter water chemistry to see if we can tweak the mercury methylation problem. And maybe we can talk about that in some of our discussions. So moving into methylmercury, we know that we need to understand methylmercury dynamics. Most of the past 40 years have been focused on inorganic mercury because that is our regulatory drivers. But we knew that we needed to take some more accurate measurements of methylmercury. So this is concentrations of methylmercury over the course of a day. And this is at an upstream location, East Fork kilometer 16.2 and East Fork kilometer 5.4, so 2 different sites. This is dissolved methylmercury concentrations, and you can see that there's definitely a diel pattern. So dissolved methylmercury concentrations decrease at night and increase during the day. This is in contrast to what happens at a number of other sites. We know that methylmercury is produced during the day because it's produced by algae and periphyton that need light. But methylmercury concentrations at other sites actually decrease because there's photodemethylation. But because we have like a very strong canopy, like we are not seeing that same signal. We looked at the diel patterns of inorganic mercury, particulate mercury, and we can see that we have an increase in particulate mercury at night. So that was very interesting to us. It seems like both particulate mercury and methylmercury concentrations increase overnight. And that's coincident with like a higher particulate load at night. And we hypothesize that this has something to do with biological activity. Either biological activity to remove these particles is not happening at night. So maybe clams and mussels are not filtering at night. And so the particulates are up. And so the methylmercury is up. Or fish and other things are more active at night and kicking up sediments. And so we know that there are strong cycles both seasonally and within a day that are affecting methylmercury concentration. So we know now that we have to focus on what is happening in biological communities. So biological communities. I showed you a graph of how mercury concentrations have changed over time in response to various remedial actions. This on the right-hand side is a graph of how the communities have responded. So we look at diversity of invertebrate and fish communities as an indicator. This is a very accurate indicator of water quality. And you can see that in response to various actions that DOE has taken over time, we have seen the species richness increase both at our upstream locations and at our downstream locations over time. But guess what? When we have more species richness, it makes the food web longer. There's more steps in the food chain for mercury to accumulate. So we're making the water quality better, but we're also making the mercury problem a little bit more complicated. And so we decided to look at, since we have 40 years of food web data, of community data, we decided to analyze both the mercury in fish along with the community data. And what we found was, so this was a correlation study where we're looking at concentrations of mercury in our sunfish, red breast sunfish. And what we found was methylmercury concentrations in the water were positively correlated with mercury concentrations in fish. That's not surprising. But we looked at the community structure, the invertebrate community structure, and we also found that the percent of the community that was collector filterers was negatively correlated. So when we had a lot of collector filterers in the community, we had lower concentrations in fish. And so one of the things that we're doing is to look at what the capacity is for mussels to filter mercury out of the water column. So I don't know if you know this, but East Tennessee and the southeast of the United States is actually a hotspot for freshwater mussels. They are ecosystem engineers, so they filter water and particulates over their gills to feed. And in doing so, they actually really affect water quality. But because of declining water quality throughout the Southeast, these are the species that are most in decline in terms of like extinction, really. And so we are working with TWRA. Their mission is to reseed our local streams with native species of mussels. We are working with TWRA to evaluate the effectiveness of putting these mussels out there in removing mercury, but then also doing an ecosystem service. So again, improving the water quality. So we've done a number of experiments where we looked at the filtration rates over time, the response to light and temperature, how all these things would happen. And we are now getting to the point where you'll see in the lab, we're getting to the point where we're going to do larger scale mesocosm tests where we actually look at, are they effective at reducing mercury concentrations in fish? I think I'm running a little bit late, so I'm going to skip this, but basically we're really trying to understand food web dynamics. In addition to what's happening in the sediment and in the water, we are studying mercury in fish and in the invertebrates and looking at the efficiency of transfer through the food web to see if there are things that we can do in the food web. Can we add species of fish or remove species of fish or mussels that can affect that transfer? But really the $1 million question that is I think exciting the mercury world right now is what controls mercury methylation. This is a very complicated question because there are so many different factors, water chemistry, physical factors like temperature or just total suspended solids that all affect microbial communities and mercury methylation. Now, the way that we don't have a handle on this problem. I will just be honest with you. But we know that periphyton, so periphyton are a consortium of algae and bacteria that attach to rocks and the benthic substrate of streams. In recent years, so I would say it's only about for the past decade, it is recognized that periphyton are responsible for mercury methylation in the streams. But it's very difficult. We don't have very high-tech methods to study periphyton. Basically, we've been going out and collecting rocks and taking toothbrushes and scrubbing toothbrushes to get the algae off of there to understand what is the chlorophyll, what is the species composition. But how, like from that to suggesting an action for DOE or for the city of Oak Ridge or some best management practices, there's a long way to go in between scrubbing a rock and then making a watershed decision. So we knew that we had to develop new tools to be able to study periphyton. So we started using drones with sensors to fly through the stream, through the canopy, I should say, to try to get at not only what is the chlorophyll concentration. So chlorophyll is like sort of a proxy for how much algae is in the stream, but maybe also other habitat metrics that we can use to predict when these periphyton are blooming, growing, and when they're likely to methylate mercury. So unfortunately, this was a video, but you can use your imagination. The drone looked like. But this is really, so you can see the reason why we can't just use scrubbing a rock is because just in this photo, you can see that there's a lot of spatial variability here. So you can see those areas, there's green, darker areas. Those are rocks that are covered with periphyton. And then there's brown areas where it's just sediment. And then there's areas that have no water at all. So there's just a lot of variability. So taking one rock in one area isn't really indicative of the whole stream. So we have to have a better way to scale this up. This is just basically saying the same thing. In other systems, larger systems, satellite data can be appropriate, but because our streams are so small and so forested, we really need to use something that can get through the canopy. These are some of the results from habitat mapping and light penetration. So this kind of can tell us how light gets through the canopy, and so which areas might be susceptible for periphyton growth. these are our very preliminary studies, but really the next step really is to see how things change seasonally over time. How does the periphyton community change over time, and how does that correlate with methyl mercury production? So as I said, this is just like an overview of some of the things that we are doing for mercury remediation technology development. There's a lot of other things that we can get into for discussion. I've kind of gone through these conclusions, but basically the take-home message here is, because mercury is so complex, because we have high concentrations of inorganic mercury in one location, high concentrations in the sediment in another location, and then high methyl mercury in the downstream location, which is not DOE land necessarily, there isn't going to be one solution. There may be multiple solutions. We might have to make best management practices or a watershed scale approach to this problem. And so potential future strategies for mitigating mercury. You all know that there's a mercury treatment facility that's being constructed that will decrease the mercury flux out of the Y12 facility. But as I mentioned, there's a lot of diffuse legacy sources downstream from there. So it will be effective in the upstream location, but there's a lot of sources downstream of there. There are things that we can do because we've identified that there are targeted areas that have elevated concentrations of mercury. So if we can do anything to stabilize the banks there, again, these are quick wins that could be cost effective and really effective at reducing the mercury flux. Again, watershed scale recommendations. So again, what knobs need to be turned to decrease mercury methylation? These are basic science questions, but we are trying to ask these questions and address these questions in a way that can be applied at the site. Again, things like, are there things that we can do to decrease the flow or modify nutrients, algae, light, habitat? And then, as I mentioned, modifying the food web, potentially using mussels to introduce some kind of ecosystem service and reduce the mercury concentrations. I obviously did not do this work all by myself. I am standing on the shoulders of giants. First and foremost, I know many of you knew my mentor and predecessor, Mark Peterson. The bones of this talk came from him, so I definitely need to acknowledge him here. And then I have a team of scientists that work with me. We've had support from Y12, from DOE, from UCOR. I basically was plagiarizing from a lot of people in this talk, but just to give you a summary of everything that we have here. With that, I will take any questions.
We will start with the board members in the room. If you have questions, just raise your son or your name tag on the side and we'll start. There's no reason for you to even do that. Go ahead.
I'll play by the rules. Basic biology. What's the advantage of methylating the mercury for the organism that methylates it?
So there is no known advantage. I don't think that they get anything out of it. So bacteria, they get energy by.
Yeah, you don't have to go into it
Okay.
for me.
All right.
Okay.
So basically, and mercury is one of the least energetic.
I started to say.
Yes. Yeah, they're not doing, it's just happening as a side reaction.
I'm just
to
thinking,
something
can't
else.
you feed them something that would be more useful
Oxygen.
as a competitor to the mercury? Yeah, all right. That fits.
Chris.
You said that it was taken up in cells, and I see the fish and such. Is it also in vegetation? Does the vegetation take it up?
It does, but so it does. But because mercury and methylmercury biomagnify through the food web, plants will take it up to a certain extent, but it's over time when something eats that, it accumulates what is in the plant. And then over time, it's accumulating more and more as it moves up the food chain. So plants can be used as a phytoremediator. Okay. But because mercury methylation is affected by flow and things like that, if you start to plant things, you might decrease the flow. When you plant aquatic vegetation, you tend to decrease the flow. You tend to like accumulate organic matter as that those plants are decaying. And now you have a hotspot for mercury methylation. So it's not that phytoremediation is not an option, but we would have to be very careful about how we would do that. But that is something that we're talking about, like not necessarily for mercury treatment, but maybe potentially for bank stabilization, for
Yeah.
example.
No,
Do you know, are you able to disclose what two areas in Oak Ridge are of concern on the creek for the high
Um,
concentration?
So I think it's public knowledge and there's, on my,
I'll just food for thought for the.
yes, yeah, right. I forgot to mention during my talk, I showed you a picture. We actually release a report every year. So if you want details on any of this, it is open to, you can Google it, like I assume that you'll all get the slides, so there's document numbers and you can look at those. But one area, so it's the stream banks, it's not like,
Understood.
okay, but in the stream banks, I guess I didn't mention. Can I go back? Okay, in the stream banks, there's a layer in the stream banks, which is kind of buried, but you can see it, it's like, it looks black. So you wouldn't necessarily be exposed to it, like if you're walking in the floodlands or anything. But one of the areas is like around kilometer 23 behind the NOAA building. And the other area is around kilometer 15, which is where the old Burners Market used to be. I don't know if that means anything to you because you look young.
Did you say that they had a plan to remediate those two areas specifically?
They actually were remediated.
Yeah,
Were they?
One of the very first remediation projects that DOE did under CERCLA was to go out and remediate those two sites.
I'm assuming the levels are still somewhat elevated, although it's.
In the areas that they were remediated, where the floodplain soils were removed, those are down to almost background levels because those backfilled with clean soil.
Gotcha.
There are areas that were below the cleanup level that still had elevated levels of mercury.
Yeah.
Actually, if you look at the number of samples that they took, it's in the number of samples that were analyzed are the tens of thousands all along the creek.
Thank you.
Yeah, I forgot to mention that when I showed that picture, the stream bank, it's like several feet down.
The area
So
underneath.
it's not like.
Hard to
surface.
disturb.
Yeah.
Understood.
Christine.
So the sorbents that you deployed, can you explain a little more? Because I couldn't really see from the distance
Yeah.
of what was in the picture of what that looks like and how do you disseminate those?
That's a very good question. How would you disseminate those? So the different sorbents actually look very different. The particle sizes are different. How they behave in water is different. And so the way we did it in this study is we had to contain them somehow. So we put them in a porous bag and like we put them in like a mesh basically so that organisms couldn't get to them and like spread it around. And we just deployed them in the soils. But then if you actually deploy them, would they be mixed into the sediment? Like those are more, after we look at the efficacy, then it would be, how would you apply these things?
Thank you.
You'll actually get to see samples of that. Oh, you can't come to the lab.
No, I'm gonna miss.
Send you pictures.
Does anybody else in the room have questions? Rachel?
I have a question about kind of this network of knowledge that you're building out and other remediation tactics. What have other places done that has been successful?
So yeah, that you asked the question. So the South River Science Team, they are a network of scientists. There's a contaminated site in Virginia, the DuPont site, right? And they're about 10 years ahead of us in terms of where they are with remediation. So we are active in those discussions. We go to their meetings and we actually are collaborating with some of the scientists. We get some of the students from there. Their system, the river is larger than ours, but it's a similar site. So we've actually used some of the same sorbents that they were using. We've taken some of the same approaches to some of the things that they're doing at their site. But we use them as like a guide for what worked for you, can it work at our site? But we have some, like I said, there's some challenges at Oak Ridge that are not exactly the same, like that layer and the stream banks, they don't have the same. Mercury is not speciated the same way at our site.
I have another one. Okay, so you talked about phyto remediation. Is there any ongoing studies with like using mycelium as particular like ecosystem services of mycelium?
We have a whole group of biologists that are looking, but not, I don't know that they're looking at it from a mercury perspective. That's an interesting question.
Christoph?
You mentioned, and it was a great presentation, I fully enjoyed it. You mentioned that some of these sorbent peels were deployed for a year. Did you measure any methylation in the absorbed mercury?
So in the field deployment, we did not look at methylation because it would be difficult to kind of tease things out because they were deployed over a period of a year and mercury can be methylated and then it can flow downstream. So there's no way really to get at that question in the field. However, we are doing currently long-term lab experiments where we have bucket tests and we have sorbents in buckets and we are looking at, again, the dynamics of mercury sorbing and then the production of methylmercury. And we have worms in those buckets as well. And then so we're looking at how sorbents affect mercury methylation bioaccumulation. And I'm saying you understand that this is not just me, that work is Dr. Scott Brooks. A lot of the work that I showed today is not my own. Full disclosure.
Thank you.
But you will get to, if you come to the lab, you will get to see those experiments going on.
Thank you, I appreciate it.
Do we have any questions online?
We do have one. Harold Connor had asked, based on lab studies, how confident are you that mercury treatment facility will make a difference in the methyl mercury and downstream?
So I can tell you that we have done some work to look at what the projections would be on flow, on total suspended solids. And so we know that we basically have made projections on how effective that the mercury treatment facility will be. And we think that there should be no times that the flow should exceed what it is built for, the capacity basically. So I think it will be effective at what the goal of that of the facility is. Now, how effective is it going to be downstream? I cannot speak to that, but the goal of that treatment facility is to treat the mercury that is coming out of outfall 200, right? So obviously if we're reducing the flux out of the Y12 facility, that's reducing mercury that is going downstream that would then be available for methylation. I can't really speculate to what it would do for methylation, but anything that we can do to decrease the mercury flux and mercury concentration can only help.
Do we have any other questions online?
I have seen no other questions.
Okay. I do have a real quick one before we go into public comments. So how far downstream do you all actually test for the mercury?
So we sample as part of our biological monitoring program down to kilometer 6.3, although we have just deployed a monitoring station for water downstream of that. But we also have a long-term monitoring stations downstream of the Oak Ridge Reservation to make sure to look at how mercury and fish evolving downstream of there as well. So we go down to the Tennessee River, for example.
I thought y'all did. I was just, I don't know, verify. Sure.
We sample throughout the clinch, both upstream and downstream of the Oak Ridge Reservation in Poplar Creek and then down to the Tennessee.
We do have, member of the public, in addition to public comment, does have just a question on the presentation.
Okay.
Yes, did you anticipate any impact from TPA shutting down the coal fire plants up on the mercury?
So as you know, coal releases mercury into the atmosphere. I don't know how far like I've never done a study to look at the transport of mercury from these specific plants. But again, we know that we have, for example, in the Smoky Mountains, there are fish that are above tissue guidelines, and that's from transport of atmospheric mercury. So shutting down coal plants will definitely have an impact. I don't know specifically,
Locally, it's hard to determine though, probably because so much of it come out of Y-12.
there are probably ways to get at that. But
Okay. It's something to watch probably.
yeah, probably.
Any of the guests have questions?
That's what I was just.
I think we're good for public comment. We do have one individual who wants public comment.
Who else would that be?
Then we will go to public comment.
Yours truly.
Okay, I lived in Gibson. I was a member of the board, 99-2005. I thought tonight's topic, I mean, we talked about mercury tonight, but this kind of leads into a discussion of ecological enhancement as a remedy in circle documents, I think. And we could talk about PCBs, maybe some other things, but anyway, there was a 2007 action memorandum that selected ecological enhancement as a non-time critical removal action for K1007 P1 pond. In September 22, the action memorandum was updated to change the remedy to pond fill-in to support the construction of the Oak Ridge General Aviation Airport. The airport project has had questionable public support or even awareness, except among those that colluded and conspired to plan it. Anyway, the ultimate status of the airport project is unclear, thus making the future status of the pond the same. Hopefully, the future of both will become clearer soon. As an alternative or complement to ecological enhancement, and I've talked about this with Dr. Matthews, I was the work of a researcher at the University of Maryland, Baltimore County, named Dr. Paul Gosh, got my attention several years ago when I comment on the potential applicability of, the in situ, treatments that have gone on there and the sort of since you're studying there. I think that's something we should be pursuing. Now I'll change subjects again. ORSOP should reconsider the use of the open May 8th meeting date for additional topics that have been raised by TDEC and myself. Presentation topics at local meetings of the other SSAB should also be considered. For example, the Idaho Plant Project Systems Advisory Board received a presentation from Dr. Justin Marble, Acting Deputy Assistant Secretary for Waste Management and Materials, on the EM Corporate Transuranic Strategy. I confirmed with the Idaho CAB Chair, whom I know, that Dr. Marble presented virtually. So headquarters presentation store setup could also occur in that way. Despite the title, Dr. Marble's presentation has slides on DOE legacy radioactive waste in general, mentioning the Waste Information Management System, or WIMS, which had the data I used to review claims about the adequacy of Oak Ridge Reservation's surplus waste disposal capacity. His presentation also mentioned the annual Transuranic Waste Inventory Report, which is essentially WIP's capacity assurance report. The latest one just came out, and it should be reviewed for what it says about Oak Ridge. Uncertainty about treatment options for true sludge cannot but impact Oak Ridge volume estimates for disposal of WIP. The original EIS for the true waste processing facility estimated that cementation would result in 3 times the treated waste volume of the initially preferred but later abandoned low temperature thermal drying alternative. We talked about that a little bit in committee, and I'm sure we'll be talking about it in the future. And finally, I would like to encourage ORSOP to consider having a real planning retreat before the start of the next board year. Thank you.
Thank you, Luther. Just a couple of reminders. The issue group on this is, there's only 4 people on it. Butler, Jones, of course, Miss Harriet, and Sharp. If y'all are interested in joining the issue group, please let Sarah or Shelley know, because we could always have more people. Yeah. I mean, more opinions are always welcome.
Amy, I think we do have one other question online.
Okay.
Having an issue, let me
Well, that's okay. We can.
Chat. She stole it. Mary Butler says, do we know if the methylated mercury accumulates in all tissues of the fish or just certain tissues or organs?
That's a good question. Methyl mercury is accumulated, it accumulates in all tissues, but it tends to accumulate in protein. So muscle tissue, which is really what we eat, right? So that's what we care about.
Thank you. I will call for any additions to the agenda. Are there any additions? Motion to approve the agenda.
So moved.
I have a motion and a second. Is there any discussion to approve the agenda? All in favor of approving the agenda, say aye. Opposed? Agenda approved. Are there any requests for new action items? Melissa, I'm gonna give that question to you. Our next meeting will be at 6 p.m. on June 12th on ETTP main plant groundwater remedies selections. I know, it's been a mouthful lately. This is the presentation portion of our meeting. This will give our presenters time to exit if they would like to. Do you all? We'll move on to the board business. Has everybody had a chance to read the minutes?
I read them carefully and they look accurate.
Do I have a motion to approve the minutes? Have a second? I have a motion and a second to approve the minutes from March 13th, 2023. Is there any discussion? All in favor say aye, approving the minutes. Aye. Opposed? Minutes are approved. Responses for recommendations and DDFO's report. Melissa.
We have no open recommendations at this point. Y'all are actively working on a recommendation for the budget, which you're waiting for. Other than that, the only other thing I would like to just remind you of that we will be hosting the EM Chairs meeting in September. So we are working on the venue and our dinner that we'll have, and we'll be getting information out that as soon as we get everything confirmed. That's all I have.
Thank you. We'll go to committee reports. The executive committee report, I'm going to pass to Chris because he actually chaired it for me because I was out of town and could not jump online. So Chris, I'm going to let you take that.
We met on April the 3rd, discussed the fall chairs. It's now, the date is, the 24th through the 26th. There, we discussed the new member orientation. The date was to be decided. It looks like now it's going to be the 30th.
And all current members are welcome to join that if you have availability.
And we talked about the budget, and there will be, the committee is going to have that together, hopefully by the 20th, so this month, the 24th meeting. And there were no action items.
I see that Mary's online. Mary, do you want to give us the EM?
Sure. Can you all
report?
hear me?
Yeah.
Great. Yeah, so our next EM stewardship meeting will be on April 24th, and we will certainly be prepared to discuss and vote on the draft budget recommendation at that meeting. And also, of course, continue discussion on today's topic. Today's topic, there is no recommendation being requested.
Thank you. Are there any additions to the agenda or closing remarks? The only thing that I know that we need to announce is that there's no meetings in May. Is that correct?
Correct.
And then please put it on your calendar that we will have our next meeting in June at 6 p.m. Is there anything else? Are we good? That's what I was looking at with the two of you, to make sure y'all were okay, that we didn't forget anything.
Keep us honest.
Yeah, right. Motion to adjourn,
I move we adjourn.
second. Of course, that didn't come difficult. Any discussion? All in favor, say aye.
Aye.
Opposed?
Put the hands up there.
There you go.
We are adjourned.